Hello everyone, my name is Chowdhry, and today I'll be talking about the synthesis and enzymatic separation and bacterial degradation of quorum-sensing autoinducer homocysteine lactones. This research project was conducted in the Advanced Chemical Techniques Lab, the ACT Lab, and is a two-semester long project that consists of a synthesis study, a quantitative study, and a kinetic study. The N-acetyl homocysteine lactones, otherwise known as AHLs, are a type of chemical signaling molecules known as autoinducers, which are used by gram-negative bacteria to communicate to each other so that they may coordinate gene expression with cell population density. This mechanism is called quorum sensing. The L enantiomer of AHLs have been found to be the principal type of autoinducers for the AHL class used in quorum sensing. It has also been shown that the disruption of quorum sensing in pathogenic gram-negative bacteria leads to the prevention of the development of virulence factors through the degradation of AHL by AHL degrading bacteria. One such bacteria is Mycobacterium dystacium. Current methods for the for the synthesis and enantiomer separation of AHLs are costly and time-consuming. For example, one such method may take as long as 72 hours and cost as much as $500 just to produce 25 milligrams. The objectives of this research project fall into the three parts mentioned previously. The synthesis study will see the development of a procedure that is low costing and less time consuming, and butyl L-butyl N-butyl L-homocysteine lactone, otherwise here on out known as BHL, will be produced because it is the most abundant and one of the most simplest AHLs used by gram-negative bacteria, especially pathogenic gram-negative bacteria. Then in the quantitative study, we will determine whether an enantiomerically pure or reciprocal mixture was synthesized by an enantiomerically selective separation method of gas chromatography coupled with mass spectrometry. Finally, we will perform a time-dependent kinetic study testing the ability of Mycobacterium dystacium to degrade varying amounts of AHL at different pHs and temperatures to determine the effect on its degradation activity. For the reference method used, it was chosen because it contains many common practices and methods for synthesizing AHLs today, especially BHL. First, we see that L-methionine is reacted with bromoacetic acid and reflux for 12 hours to obtain L-homocysteine lactone hydrobromide, the reactant, at a percent yield of 75%. Then the acylating reagent, butyl chloride, was obtained with the reaction of butanoic acid with ethynyl chloride, reflux for 3 hours. The percent yield was 90%. Then the BHL was obtained through the reaction of the reactant and the acylating reagent with a percent yield of 86%. And the new and this and all led to cost of $230 to produce 25 milligrams. In the new procedure, we see that L-methionine is reacted with bromoacetic acid and stirred for 4 hours to obtain a sulfonium compound. This same sulfonium compound is then reflux for 2 hours to obtain L-homocysteine hydrobromide. Then L-homocysteine hydrobromide undergoes a dehydration to obtain L-homocysteine lactone hydrobromide with a percent yield of 80%. Then the same acylating reagent is obtained. Only its the reaction is reflux for 2 hours, and the percent yield is higher at 94%. Lastly, the acylating reagent and the reactant are reacted together to obtain BHL. However, the percent yield is 85%. This only costed $20 to produce the same 25 milligrams. The HNMR data shows that BHL was indeed obtained through the new method. There are extra peaks because the BHL did not undergo flash chromatography in the new method due to time constraints. However, nonetheless, we see that BHL was indeed obtained. So for our conclusions, we can say that our method is less time consuming because our method only took 7 hours, whereas the reference method only took 15 hours. It's less costly, and our product was obtained. For the future studies, the first one is the quantitative study. In the quantitative study, we wish to used in Agilent 6890 and GC equipped with 5975 MSD to carry out all separations. And the column will be a beta DEX 225 column, which will will separate the enantiomers, as we can see in this chromatogram. It will separate the D enantiomers and L enantiomers distinctly. Then for the next study, we will take the bacteria, Mycobacterium dystacium, and expose it to different amounts of BHL, specifically 10, 25, 50, 75, and 100 microliters. Then we will expose this to different temperatures, 20, 37, 50, and 70 degrees Celsius, and then different pHs, 3, 5, 7.4, and 9. We will test the kinetics of this by testing how well the degradation goes at 12 hours, 24 hours, 48 hours, and 72 hours.